Thank you very much for the invitation. My name is Ernesto, Lasso de la Vega. I work for the Lee County Highs and Control District here in Fort Myers. But I'm a biologist that has been teaching at the universities, but I live in Port Charlotte and I play in Sarasota. Play because Sarasota Bay Watch is a root citizen organization that are really care for this bay, for Sarasota. They do not just clam restoration, we do also cleanups. We clean up islands, we underwater cleanups, we care for monofilament. So there's a lot of activities that go along with the Sarasota Bay Watch. And one of the projects that they have is the one that I'm kind of leading, I guess, is the clam project. Now, why do we do clams? Well, we started, and you might see here, and I have a few slides where they're underwater. We started with some scallops. And the scallops is because the scallop is a very, very unique organism. It's actually a filter feeder. It's an indicator, but it's very weak. In fact, I have a video here, which I want to just play it because it's kind of cute. Let's see where my mouse is. The mouse is not, the mouse is here. Let's see, roll, okay, go this way. Okay, now, click. Okay, so you can see the clam, the scallop that's on the water is this creature that you see in the middle. There. That's the scallop, I mean, just close. So this is the first clam that we started doing projects by enhancing, by providing more numbers of scallops and we start spreading it around. And it's an indicator. This guy is an actually indicator. And I have another one. Gosh, I really wanted to show the clam, but the video is gonna show some of the clam actually swimming around. Come on, clam. There. So scallops were very charismatic. It's an indicator. And we started with that by putting thousands, millions of scallops there. But they are very weak. In fact, you might see them swimming around because that's what they do. This guy, I actually filmed this, so I removed the little fuss that he has as an algae there, and then you can see he just swims away. So this is a very unique characteristics of the scallops. Whenever you touch the scallops, they just take off and they actually swim away, and that's what this guy is doing there. Swim, 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 swim. It's a beautiful thing. It has beautiful blue eyes, and if you see these guys are swimming, it's fascinating. But they are sensitive to red tide, sensitive to fresh water, to bad organisms. So we switched to clams because they're tougher. A clam can filter water, like about 20 gallons per day. A single clam can do that, so we are looking at the filtration power of these organisms. Now, the clams were there a long time ago in large quantities. Fisheries came and took them, a lot of them off, plus the environment has not been improving the water quality for the clams to survive. Plus you have a lot of nutrients who make the water more turbid. So there's a lot of connotation that come along. So the numbers of clams have been going down. So we saw this as an alternative to improve the waters of the bay in Sarasota. So we hired a clam farmer and we actually are hired to them to raise our clams, and then we move in from Pine Island, which the clams came from, to Sarasota, and we did the whole motion of that. So they can live up to 20 years. These little guys, 20 years, I have some fueling, and you've probably seen the large size, like 6 inches clams, and they are 15 years old right there. And again, the power of a clam, when we're talking millions. Now, Sarasota Bay Watch, let me show you in this video. This actually is an actual video of the activity. Loud.
On the right side of the keyboard is the volume.
Okay, I got the yeah. So here, let me play it. Oh, just cut the volumes down so I can actually narrate when you guys are looking. So this is the actual activity of Sarasota Bay Watch. There you go. So we harvest those clams from Island, put them in this refrigerator truck, donated by the bud company, and then actually carry all those clams in baskets into barges where we're going to be putting in boats and later on distributing. We're talking 60,000 clams in one of these, not in one basket, I mean hundreds of the basket, but about 1,000 clams in one of the baskets. Volunteers provide their weekend just to do a Saturday. There you go. And we are going in boats and dumping it in certain areas. So I saw this activity and I was like, wow, we gotta do this in a way that we can measure success. And what is success? If these guys start reproducing. Because we're not gonna be doing clam restoration for the rest of our life. So we put the clams there, and later on we come, and these are the clams already freshly put in there, and you see them, poof, they're digging themselves into the mud. It's clamtastic. We had to put a little joke in there. So where are we putting the clams? That's another thing. We took maps from Note Marine that they did an analysis of all the clams in the bay, and those little squares that you see in this black and white map is actually dots where the clams were present there. And then we match these mappings of the seagrasses, so we don't want to put the clams on top of the seagrasses. So what we have done is selected areas where they are missing clams, and so we can put the clams, and they were originally there. So we explore those sites, we go and decide to put a 50 by 50 meter square to put the clams in that location. Now, why 50 by 50 meters? In fact, let me see the next one. Yeah, okay, in addition to that, we had to see that the soil, this is a bad soil. It's all organic, black, it's anaerobic. Clams doesn't like that, they don't like that. And then they see also drift algae, they don't like that because there's a lot of oxygen depletion that is happening in it. Where you find a lot of nice, sandy, muddy, that's where they like to bury themselves. Plus, if I find some predators of clams, that tells me the predators there, the clams are there because they're gonna eat them. So that's our indication. So we go and find those places and we said, okay, let's take a 50 by 50, and that's what we do. These areas has been already established. We put buoys like this one, and we put them so the boaters can come, and in that 50 by 50, we're gonna be distributing those clams. Now, why 50 by 50? Because at the numbers of clams that we're putting there, they're gonna make the density of 25 clams per square meter. Well, what's the important number there? Because at that density, the clams will be able to produce sperm and eggs, and they will be able to fertilize. It's external fertilization that the clams do. So once they have fertilization, babies will come. And that's what really we're looking. I mean, the filtration, granted, is part of the plus. But the reproduction is what I'm really looking for. So in my perspective, now we're just finding out how many clams are there. If they survive, good. Next year, if I find the survival is good, bingo. But if I find babies, super bingo. That's my really goal on this. So this is a little video I have about where we have put the clams right now in Sarasota. Of course, this is a secret. I don't want people to reveal this because poaching can happen. Okay, so this is right there in some of the locations. Again, all of them, the yellow dots that you see there, they're all squares, 50 by 50. We found locations, the clams are not there, we put them in there. We go around here, there's another spot, we put them in there. So in the future, again, we're gonna be going back again in those spots and find out if the survival is good. And if it's good, well, okay, great. And if it's not, we also learn from that. So it's not just like, yeah, good results are good, but then bad results are also good, so we learn from that. Next. I'm going to be moving. We started in 2017, putting about 200,000 clams. We made a goal to put about 1 million clams. So 1 million clams has been bought by Sarasota Bay Watch. And we did this fundraising. It's called Scalopalooza, because we used to call it Scalopalooza all the time. This was a very neat experiment that we put in that hall. So there was a bunch of people there in that hall, and we put it was a dinner celebration, and then we put these jars with a lot of algae there, so it's turbid. And then the students came and they dropped like about 4 clams on each one of those. In 1 hour, they crystal cleared the whole jar. People saw that, it's like, oh my God, I believe in this power of filtration. So they contribute money to us, we raised fundings, and we were able to use those funds to buy clams and to continue this process. Now, again, I need to validate this because it's not just buying the clams and put them in there. No, we need to validate that. Okay, so here's some of the clams. I want to briefly show you that these guys, that's their habitat, but in about an hour, maybe half a day, they will bury themselves, so you won't see them like that. This is freshly put down, so they're gonna dig themselves in, and you're gonna see them, well, not here, this is not video. Some people ask me, how you can measure the age of a clam? So each one of these ridges that you see there doesn't represent a year. You have to cut them, and if you see berries here, there's some groups where they're very tight together, and there's some open spaces. Well, the open spaces means that they grow fast, and the tight ones, it's just like the rings of a tree. The tight ones is that they didn't grow so fast, so fast growing, short growth. In fact, that's how every year they're showing some rings and rings. So that's how we count these open spaces and dark spaces, and that's how we determine the age of the clams. Although you can tell the age of the clams by just looking at the length of the clam. Pine Island is where we put the clams and we raise them. There are clam farmers there, and these are nice shots that I want to show you. A clam farmer, he has a lease. He put the clams down there, and he put in a net, so he protects from predators like the stingrays. There's a conch. This is a whelk conch, a lightning whelk conch, and he comes and he chews on the clams. So he protects them from these guys. Then we raise them to about 2 inches, and that's where he pull them out. This is a farmer, the hatchery man there, and then another farmer, and they're all going to be pulling those nets out of their boats. Oh, and they have a vacuum, so they vacuum from the mud, put them in bags, and the bags are going to be all clammed up there. There's the clams in the bags. Then the bags will pull up, put them in the baskets, and put all the baskets, and they move the basket, the boats, right to the dock. And sometimes we have San Diego Cativa Conservation Foundation comes and help us carrying those baskets. That's a lot of baskets. So we carry them all to the dock, and in the dock we have the refrigerator truck, and we put them in there. And then we transport them all the way to Sarasota, where we have nice volunteers all ready to put the clams in the boat, in the water. Voila! There's Rhonda. That's my executive director, the one who's so excited. So that's how the trip goes. That's a lot of work, but it is great work. This community lives that. They know what they're doing. They feel very powerful about it, because this is a little thing that can be done by citizens. So they asked me, like, the water management district, no, the National Estuary Program asked us, so we don't believe so much on the power of the clams. I said, well, okay, you don't. What do you believe? We can talk to the wastewater treatment plants and the county to enforce these laws and stuff. Okay, you can do that. We can't. We do clams. We can raise money, we can buy clams, we can put the clams out there. Leave us to do something that we can do. And that's what we're doing the clams. I mean, of course, the other stuff is just, politics and talking to the big guys. One last thing I want to bring up is that because we saw the future, we cannot just be bringing clams from Pine Island all the time. Come on, we can put a lease in Sarasota, which we did set up this area here, and we put a little square there. This is a 5-acre land that we asked for the Department of Agriculture to let us grow clams in Sarasota, so we don't have to transport them back and forth. So we have the clam lease. This is the first restoration clam lease that has been approved by the Department of Agriculture for a citizen group to raise clams for restoration. I'm so happy about that because we just got that approval, and that means that we can grow the clams there, and then once we harvest those clams, we can move them where we want in Sarasota. So it's a big achievement. So we're going to be raising clams for 2 years and then moving them around. There's an interesting fact here. So the clam filters algae from the water. So that is removing nitrogen in the form of algae, and then it assimilates into the clam. So if you harvest the clam, you're removing nitrogen from the system. But there's another interesting fact. The clam poop. And the poop has transformed the algae nitrogen into feces, called biofeces. And that biofeces can be absorbed by other organisms, such as seagrasses. And there's another impact. The carbon can be also calculated, how much the carbon has been absorbed by the clams to form the shell. And they're working on those numbers, to make it credits. Credit for nitrogen, credit for carbon. But the one I'm really excited is about the seagrass and clam, what's called biosymbiosis. Because the clam poop, the seagrasses like that type of nitrogen. Seagrasses will be growing, enhance the growth for the seagrasses. So we have seen in sites where we put our clams, seagrasses start colonizing that. My goodness, that is a great, because now we are enhancing the soil. We also are enhancing locations where the seagrasses are going to be growing, of course, and the rest of the animals will go along with the seagrasses. That is a big one. We're doing an experiment there. We have plots with clams only, plots with seagrass only, and then mix of those two to see which one grows the best and what are the effects. All that's kind of a research. The Gulf Shelf Institute is helping us to do this kind of research, and we provide the site, we provide the clams. Let's do research, because we believe on the data that comes out of this, it's going to be important for future generations to know this. I'm getting all excited. Let me calm down. Okay, there's a lot of wording here, blah, blah, blah. What are the lessons learned? Well, we look at stuff from Mode Marine. We talk to Mode Marine scientists. We learn a lot from that. Clam farmers, we learn a lot from that. But I'm looking for future things like DNA. If I find a baby clam, and I had the DNA of the hatchery who gave us those clams, and I match those guys with my baby clam and the hatchery DNA, wow! Do we have a cherry on top of that sundae? It is awesome. So that's kind of like the evidence that I want to finally have at the very end of this whole project, like the DNA matching of this stuff. And what else we have learned? Well, people like the idea. Utilizing the clams for algae reduction, combining clams and seagrasses, opportunity for students to do research. Now other organizations like Sanibel Captiva Conservation Foundation, they want to do the same thing. I said, do it! We got clams. 20 million clams. Gallons a day can be filtered by 1 million clams. And that's our kind of like a final slide there. And that's it. I don't have any more. I would like to hear questions. Do I want to go over? Thank you. I want to hear questions and comments. Yes?
Okay, I have a question. So in the Sarasota Bay, are you referring to clams there because they have been clams before, or is it because you want to improve the water quality? I'm asking because sometimes when you introduce new organisms there to an ecosystem, it can also turn the other way around, especially when it comes to seagrass, like we've seen in the Mediterranean Sea.
Correct. In fact, there's a lot of answers that I want to give from that question. Great questions. Okay, first, the animal that we put in there is the native southern clam. There's 2 clams that are being, the northern one is Mercenaria, and that's the scientific name. The one in the south, where we are living, in Sarasota, is called Mercenaria campechiensis. From Campeche, so all the way down in Yucatan Peninsula, that's where the range of that. So we're using the southern clam to respect the genetics. Yes, so we're introducing them. And what was the other question? Oh, so if the clams were there? Yes, they were. But the environmental conditions, also overfishing, has declined the populations. And that same story happened in Indian River Lagoon. By the way, on the other coast, They harvest clams miserably bad. And so they didn't leave any crop, any adult to repopulate. No, they just took it because they were there. I'm taking all. And then later on they realized, where's the bait? How are you going to find any more? Because you took all of them. You need to be something sustainable. Now, what was the other question there? No, that was it?
I brought up the example of the seagrass, how it grew in the Mediterranean Sea, so that it was actually a negative effect at the end.
Actually, there is a lot of agencies, Fish and Wildlife, and the Department of Environmental Protection, and the Gulf Shelf Institute are working with us to do this experiment with seagrasses and clams. So we have a cloud of researchers who are looking for those answers, because again, we want to answer things that are going to be used for this restoration. And we believe on that. In fact, that paper that I showed you, this is from North, that they were doing that and they were proving that already. So we're going to prove it here in Sarasota. Yes. Thank you. Yes.
What's your perspective on the water issues that Lee County supposedly is having? The water issues plus your perspective on blue-green algae in Lee County. What do you think about those two items?
Well, the algae, so it depends on what is the source of the algae. I mean, if the algae is freshwater algae, these clams that live in saltwater. So that is not right there. For the clams to do work, they should be in saltwater systems. So we want to kind of like work against pollution that is in saltwater systems. So once you have runoff from the creeks and the rivers and everything just dumping into a bay or a harbor, then that salinity is going to be, I mean, all those nutrients from that water that's coming is going to enhance other type of algae that is microscopic, and that's what makes the turbidity in the water. And then these clams will be working towards that, against that. And that's how we see our model on that. But I don't think it's just the clams. It's all kind of different filter feeders. Because there's oysters, there's the scallops, there's other creatures that I haven't even mentioned, crossbar, Venus, sponges. Everybody's filtering there. But we pick clams because we can handle the clams. All I'm saying is that we putting the one that we can handle, everybody else is going to be benefiting. In fact, one question that somebody asked me earlier, so if we put the clams and we have some stingrays who come and crunch on your clams, leave them alone, yell at, have them, our clams, who cares for our few clams that the stingray will take? Let them have it. This is part of the ecosystem. Same if I see a whelk eating my clam, let them have it. He's part of the story. Cannot just be, oh, no, don't. In fact, I argue with Mote Marine, they came up with a strategy. Put some bamboo shoots. So when the stingray comes, he cannot like those bamboo shoots, he won't be eating. Let them be. You know, stingrays also ate. So what if they take a few ones? 60,000 clams, we can spare a few. Sorry, I got excited about this clam story.
What about human culture?
What about human what?
Poachers.
Oh, that's a big one. So those maps that I show, I try not to, because people will see that and they will say, aha, how many you have in there? They can go. In fact, so we're talking to Fish and Wildlife and some other policing so they can take a look on the locations that we're putting so they know. So if somebody's lingering too long in there, yeah, that reason. So we don't mark anything. We don't leave any mark. The GPS is all that we have is the GPS locations, but we keep it as a secret for them.
So does FWC patrol the area?
We're gonna start telling them where the locations are. So if they see somebody anchor in there, yes, come and talk to them.
They better, because where I grew up,
Uh-huh.
they had clam beds, and they had markings for the clam beds. And if any boat got near those clam beds, you heard the shotgun blast before you even shot it.
Hey, that happens. In fact, in Cedar Key, whoa, that's serious stuff.
Yeah.
Pine Island, serious stuff.
Yeah.
Yeah. No, but yes.
I'd just like to say I really appreciate your enthusiasm.
Sorry, continue.
I would like to say I really appreciate your enthusiasm.
Oh, thank you so much. Yes, it's a passion. When I first started with this group, I started asking questions like, okay, so how are you gonna prove that this is actually working? Well, you're gonna answer that question, Ernesto. So then here I am, doing all the scientific stuff that I say, like, okay, let's do it like this. Because in the first videos that you saw, they're dumping the clams. I said, dude, that doesn't make any sense. Put it in a certain area, we can come back and measure that. Density, the population, measure, growth, survival. That's too complicated. Do it! Do it, because if you don't have that data, then you're not proving anything. And that's what they're failing in that. So they're all upset, like, just do it. It's gonna be a lot of hard work, but the answers will be there. And then they will see, okay. And if the answer is a bad answer, you still want to reveal that. Because maybe we had to put more clams, more density than the density that we thought it was good. Maybe you had to enhance the density. Or, maybe we're feeding the stingrays. So what? No, but I have a feeling, because this has been going in other places, like Indian River Lagoon, north in New York. They already have way years ahead of us. So we're just learning from their experiences, and we're just applying it here. Yeah, just look and read. Yes, sir.
For the manatees, can you do it?
Well, there's 3 types of seagrasses. The one that the manatee likes is called Syringonium. It's a round, like a spaghetti. That is not the one that has been colonizing. However, we can start playing that game with this kind of a research. Who says that we can just have the Thalassia, which is the turtle grass, there with the clams, which is the slides that I show with the clams and seagrasses? Well, why don't we try other type of grasses and then see how the interaction works, which one works the best? It's all now a matter of research, and start looking into that. There's a lot of thesis opportunities for students to start doing research with these things. Once we plan this and say, okay, now start collecting data, tell us what you found. We helped one group of students from New College, and she made her dissertation, and she was so happy. So all the 4 locations that she selected is because places where we dump the clams. Okay, so the question was, which one of those grow the best? So what she did, she put 25 clams in a bag, and every month she went and collected the bags and measured them. She determined which was the best place where the clams grew the fastest, which is the place where we choose for our lease, because our lease is gonna grow clams, and that was the best place to put the clams for their lease. My goodness, I was so happy, and I told her, because of your work, that's what gave us the answer for a better management of this project. Gosh, I'm not excited. I need one. Damn, clams. And you can eat clams. Don't worry about clams. Are food. They're great.
Yeah.
Do you know that the clams okay, so the clam farmer has different sizes. The big size, the medium, little, and pasta. The tiny pasta. Why pasta? Oh, because that's what the chef called him, pasta. All right. But then the cost of the clams, the medium size and the medium size, the medium size is the best value, because that's the people can eat it. The large size, forget it, it's too chewy. That's for clam chowder. It's like that. So the price drops when you have a big clam.
That's true.
Yeah, and they're like, oh, gosh, yeah. But we like the big ones because they will be producing babies and blah, blah, blah. And they can survive better. The other thing, the stingray comes, and who's gonna pick? The big, hard to crash, or the little one? Little one. So she goes and She fluffed the dirt, and then once she get it, crunch, crunch. Mode Marine, which is another research that they came with a sound acoustic receiver, and they're working with us because they asked me, oh, okay, so where are you gonna put the clams? On this place. So they put a receiver a week before we put the clams, so they listen, nothing. When we put the clams, crunch, 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 crunch. I know, it's so sad. But it's like a very unique sound because it crunch, 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 crunch. God, it hurts my feeling. But the Mode Marine is doing a research also on the predations by stingrays. And you can see those guys, moving because they have put tags, and they're moving just wherever food is. Okay, more research. goodness. Yes?
Do you plan on measuring the water for clarity or for better quality?
That is a great question, and I want to put that to the test because, okay, and this is anecdotal, but I want to prove it. The clam farmer told me, and I saw that by going and swimming in his lease. In the area where he has his clams, the water is crystal clear. In fact, you saw some, oh, thank you. Yeah. So you saw some of those pictures that were very clear. It's because the clams are filtering that. So I want to go and take water samples and then in this 50 by 50 and then go outside of the 50 by 50 in a very calm day so I don't have currents. So test that. Do the inside of my 50 by 50 and then the outside just to see turbidity by algae, chlorophyll A. And nutrients too, because that's another one. I want to know, you know, the poop, if it's releasing nitrogen and how much nitrogen. Oh my God, yeah, I'm gonna get excited.
Does having a healthy bed of clams change the microbes in the ocean as well?
That's what I was gonna tell you. There's an important factor is the microbiology that works in that layer. Everybody benefits, but we don't know who is there. And that's like one of those questions that scientists are like, Eventually, we're going to find out that it's all microbes. Yeah, this world is going to be just microbes. Anyway. Yeah, did you know that so we have bacteria, and you know this, bacteria in our gut, and they can control our mind because they send in signal. Oh, I want that pizza. No, Ernesto, you don't want that pizza. Yes, I want that pizza. But what about the salad? No, pizza. And they're just controlling yourself. So I believe the power of the microbes, is there. Gosh, I'm so excited. Right. How much of my time? 8 o'clock.
Yeah.
What do you do in terms of a hurricane? Are these clam beds, are they hurricane proof?
No, they're going to be covered by debris, but the clam bury themselves. So they stick their siphon. There's 2 siphons. One, they breathe, they suck, and the other one, poop. So the 2 siphons, and they say they can actually bury themselves for about 3 inches down in the mud. So we come with rakes, and we put our square meter, and we go rake until we pull them all out. Then we measure them and return them back. Because
you don't think they'd be blown away or swept away with the water, by the water?
No, I think they've been pretty solid on the. It's mostly motion of water and wakes and all that stuff. You barely feel it down there. It's mostly in the surface. 12 feet is the depth that we put in these guys because we don't want the normal guy who comes and start digging around. No, you have to dive 12 feet down in order to find our clams. And that's part of the strategy that we have. Okay.
That's interesting. The best refuge for a hurricane would be underwater.
Yeah,
right?
absolutely. Down there, woo, and then it's solid down there.
Best place to be.
Yeah, but then you need air.
Thank you.
No, but I'm excited about this. This group has been phenomenal as far as having a vision. And we started with a few group of people, biologists, who said, hey, let's do something. And then they decided to first start with the scallops. And the scallops, they die. 2 years, nobody remembers scallops. But they're indicators, clams, do the clams. And that's where now oysters, somebody told me oysters, but the problem with oysters is that they're stuck in their place. So you cannot move them so easy. And you can do the beds and put the oysters, and some people don't like the oysters when they grow and they make an island and then the boats ran onto that and they peel the whole boat. So clams are probably the easiest for us to handle. And I think that we stay with that. Sweet! Thank you so much for listening. I appreciate the opportunity. And if you have any questions, I got cards, Sarasota Bay Watch. Would like to have any comments, or if you want to participate in any activities, let us know.
Do you have a couple minutes to talk about the hyacinth control?
The hyacinth? Well, if you have a question, yes.
Explain what that department does.
Okay, very quickly, this was created a long time ago with mosquito control. So mosquitoes have been controlled by usually chemicals and biological ways to do that. Hyacinth came up because of navigation. The Caloosahatchee was choked with this exotic from South America. And it was brought here because it's beautiful flowers. And they brought it at where was that? The Cotton Fair in Louisiana in 18 something. And they just brought it and they took it because everybody thought it was so pretty. And it ended up in the St. Johns River. And it from there spread all over the world. And from the Louisiana Cotton Fair, went all over the world. You can find hyacinth from South America in India, in Africa. It's like, what? In Lake Victoria you have, hyacinths there. So the problem with it, there's no predators against the hyacinth. So they're gonna grow, and they grow fast. So what we do is we use mechanical, biological, and chemical control. So we integrate all three. It's not like you just spray the hell out of it. No, you have all kind of tools, and we doing that. And the biological is a bug that comes and eats and stress the plant. But mostly has to be chemical because it's unaffordable. Yes?
Does water hyacinth help purify the water though?
They do, but their rate of growth is so fast that they end up having an anaerobic condition in the pond, canal. They can choke a pond and then all the fish will die from lack of oxygen because this thing covers that. Plus, the mosquito larvae can live underneath, and that's what we don't like. So hyacinth and water lettuce, those are floating plants. We want to definitely eliminate them for navigation, also because they hide mosquito larvae in there. And then the roots of the hyacinth and the mosquito, the little gambusia cannot come and eat the larva because the roots are so, No,
What do you do with the hyacinths after you harvest them?
We thought about doing some mulching, but it's very expensive. It's 90% water. The fiber that comes out of that is so hard. And again, and you cannot even make compost because there's so much water there. And then also the other thing is that, again, it's too expensive to harvest all that and process. You better off just, eliminate it with a chemical control. And then unfortunately and people say, well, bring the manatees so they can. No, manatees will not eat that stuff. They will eat something else and they will leave the hyacinth alone. So, and you cannot harass the manatees like a cow. So you go eat the hyacinth. Eating his manatee grass. All right, I think I over 5 minutes. So thank you so much. If you have questions, let me know. All right. And this wrapped up the night. Yep, all set?
All set, thank you.
All right,
very
you're
much.
very welcome. Well, thank you.